Hi, my name is Danica Palmer, and today I'll be talking about how ATP moves throughout the cell membrane under fluorescent light into cancerous tumors. What is ATP and how does it work? And, you know, so ATP is primarily produced in the mitochondria. Everybody knows what the mitochondria is. It's the powerhouse of the cell, and it's during cellular respiration. And this involves a multi-step process that converts glucose and oxygen into a usable energy. And it's a 3-step process, okay? 3 main stages of this multi-step process, which is glycolysis, that starts in the cytoplasm, and then the glucose is broken down into 2 molecules, producing a small amount of ATP and NADH. And then it goes into the Krebs cycle, which is then further broken down, releasing carbon dioxide and generating NADH and FADH2, which then carries high energy electrons. And then it goes into the electron transport chain and the oxidative phosphorylation process, which then includes the high energy electrons from NADH and FADH2 that passes through the protein complexes in the electron transport chain. Then this drives the creation of a proton gradient across the membrane, which then powers the ATP synthesis to produce ATP. So you're probably wondering, what is ATP and how does it work? So I've already kind of explained to you what it kind of is and how the process works, but how does it actually work? So it's referred to as the energy currency of the cell because it provides the energy required for various cellular processes. It's the movement throughout the cell, which is essential for maintaining life and facilitating metabolic activities. ATP is primarily synthesized in the mitochondria. What is that? The powerhouse of the cell during cellular respiration, like I said, in that 3-step multi-process where glucose and oxygen are converted into energy. And the ATP movement begins with its diffusion out of the mitochondria into the cytoplasm. And since the ATP is like super small, it's a super small water molecule, water soluble molecule, that can move freely within the aqueous environment of the cytoplasm by simple diffusion. So this really touches on my actual research. How does ATP and fluorescent light work together? The whole point of my research was to see how ATP moves throughout the cell membrane under fluorescent light into cancerous tumors. And so these ATP molecules are tagged and they're introduced into a living cell cultures that reveal remarkable insights on how energy is generated and transferred. These markers allow microscope techniques that allow the microscope to track individual ATP molecules as they move through the mitochondrial inner membrane. And it shows how they interact with the ATP synthesis complexes and navigates the intricate electron transport chain. And these fluorescent ATP tracking represents a groundbreaking technology in cellular biology that allows researchers to visualize the intricate dance of energy molecules within living cells. These specialized fluorescent probes, typically designed with fluorophobes like green fluorescent proteins GFP variants or synthetic fluorescent molecules, emit this distinct light wavelengths when excited, and then it enables this high resolution of imaging of ATP dynamics, which is super cool. And lastly, by attaching these fluorescent molecular tags to ATP molecules, scientists can now observe in real time how these critical energy carriers move and function across various cellular compartments. So you're probably wondering, why is cancer research been put in there? If you haven't been listening, my whole research project is how ATP moves throughout the cell membrane under fluorescent light and seeing how they travel throughout these cancerous tumors. So that's why cancer research is in my thing, because my mentor is solely focused on cancer and how, ATP moves throughout the cell. So cancer research has leveraged this fluorescent ATP tracking to understand the metabolic reprogramming of tumor cells and how cancer cells modify their energy production and ATP distribution. So now researchers can gain insight into metabolic adaptations that support rapid cellular proliferation. The fluorescent markers allow researchers to observe how mitochondria and cancer cells are restructured, showing altered formations and modified electron transport chain functionalities that enable these cells to generate energy through these unconventional pathways. These observations help scientists develop potential therapeutic strategies targeting cancer cell metabolism and potentially offering new approaches to interrupting tumor growth. The answer everybody's waiting for. What are the results on this thing? How does it work? All this other stuff. I don't have the results because I was unable to get into the lab, but instead I provided insight on what I'm specifically researching and examples of how ATP moves throughout the cell under fluorescent light in cancerous tumors. And we went through how it works, what ATP is, how fluorescent light works, and how they tag team with ATP, and how this benefits cancer research. Lastly, this is it. This is my resources and citations, carefully curated, carefully sorted through. I hope you enjoyed this presentation and maybe it intrigued your interest of how ATP moves throughout the cell under fluorescent light through cancerous tumors. Until next time, bye bye now.